Good evening, everybody, and welcome to the 9th Annual Cape Perpetua Land-Sea Symposium. Grab your favorite drink of choice and enjoy the evening with us. I'll go over the agenda really quick. We're going to be hearing a welcoming address from Representative Gomberg. We will be hearing a keynote presentation from Mark Hixon on coastal connectivity, a web of life. Our intermission will be Tidepooling with the Oregon Coast Aquarium. And then we will have 3 short 10-minute presentations. The first one on determining salt marsh restoration outcomes using focus groups and ecological data with Julie Gonzalez. Our second one will be the role of Oregon estuaries in the recruitment and early life stages of rockfishes with Scott Heppell. And our third one will be mariner and fisherman perspectives on ocean changes with Captain Pazar. And at the very end of the event, we will be doing a panel discussion. And so please, throughout the event, as questions occur to you, please put questions into the comments and we will gather those and use those for our panel discussion at the end. Our plan is to adjourn promptly at 7:30, if not sooner. And we're going to be leaving you with a video about the historic 804 Trail. I want to share a land acknowledgement. The Cape Perpetua area landscape from Yachats to Florence is the traditional territory of the Siletz tribe and Coos, Lower Umpqua, and Siuslaw tribe. And we want to acknowledge the tribal governments and their roles historically and today in taking care of these lands. And I'd like to introduce our event organizer this evening. My name is Tara Du Bois. I'm the communications coordinator for the Cape Perpetua Collaborative. And it was a privilege to organize the event with a variety of partners. We have Joe Liebezeit. He's with Portland Autobahn. Paul Engelmeyer. He's also with Portland Autobahn. Bree Goodwin. She is going to be helping out with tech behind the scenes. And we have Charlie Plybon, and he will be our MC through the evening and keeping us rolling along. And with that, I'm going to turn it over to Charlie to kick off the event.
Thanks so much, Tara, and welcome everybody. I'm very excited for our 9th annual event here, and I'm going to be turning this over here to Representative Gomberg, my coastal representative here on the Central Coast, who's going to give us a little bit of an introduction and welcoming address about some of the good things that's been going on in Salem and what's to come next year. So I'll turn it over to you.
Well, thank you so much, Charlie, and thank you, everybody. It's so good to be with you tonight. I'm so very pleased that I could join you. I'm so very sad that I can't be joining you in person, but most of all, I'm so very, very grateful for all the work that you do each and every day to benefit our coast and our coastal communities and the people that live here and the people that are going to live here in the future. Well, I wanted to take just a couple minutes and share a bit of information with you. And first of all, for those of you that live in North County, or for those of you that may be visiting North Lincoln County in the next few weeks, I really want to encourage you to stop by the Lincoln City Cultural Center in the old D-Lake School. And on the lawn there and inside the building, you will see a remarkable exhibition called Washed Ashore. And what you will see are large artistic sculptures made entirely out of plastic beach debris. Amazing pieces of art made out of horrible pieces of trash. And the message here is that so much of this stuff washes through our streams and into our oceans every single day. And as you all well know, this stuff never goes away. I mean, it breaks down into smaller and smaller parts, eventually working its way into our waterways, into our fisheries, into our food stream, and eventually into each and every one of us. Charlie and I took part in a gathering with Senator Jeff Merkley at the Oregon Zoo a few months ago, when it was a lot sunnier than it was today. And the senator mentioned that each week, every single one of us ingests about 5 grams of microplastics. 5 grams. That's about the amount of plastic that's in a typical credit card that's in your wallet or purse right now. That's how much plastic we ingest every day. And that's why it's so very, very important that we continue to work on minimizing the amount of plastic that goes into the trash flow and making sure that we can do something about trying to get it out before it gets into each one of us. I know that you're already aware of all that stuff. I mentioned it tonight so that you know that I'm aware of it. Well, a lot going on in the last legislative session, but a couple of highlights that may be of particular importance to you or interest. One is that we worked hard to pass a bill dealing with cable landings on our Oregon coast, putting some safeguards in place for when transatlantic cables come ashore, and they come ashore through state lands, which is the undersea portion of the territorial sea. Then they come through our state parks, which is our beach, and eventually they get onto dry land, which is controlled by our counties. So there's a lot of permitting in place, but when something goes wrong, there's not a lot of safeguards. And if you've been following the story up in Tierra del Mar, when Facebook brought their cables ashore, we saw that everything that could go wrong did go wrong. And we're taking steps to mitigate that and make sure that the next time a cable comes ashore, that we've got some tools to mitigate the damage and to correct the problems and to really minimize the chances of these kinds of things going sideways as they have in the past. I'll also mention that we were able to achieve $1 million for the Oregon Science Trust, a board that I serve on, which is designed to provide grant dollars for ocean research. Very pleased to see a breath of life being breathed into that organization so they can move forward with the opportunity to look at what's going on in our oceans and what we can do about it. And then finally, another piece of news is that the Hatfield Center has opened their marine science building in Newport. And if you haven't had a chance to stop by, you really should, not only because it's a beautiful building, but because it's a remarkable piece of architecture and is in fact the leading example in the world, not just on our West Coast, not just in the United States, in the entire world of engineering and architecture and planning to create buildings in the inundation zone that are tsunami resistant and provide a vertical evacuation point. Big ramp along the outside of the building so that people who need to get to safety in the moments after an earthquake and before the tsunami can get up to the roof. It's a remarkable piece of engineering and architecture. Take a look at it, but also go inside the building, take a look at the ocean-oriented artwork that's available there and the classrooms, and know that this is rapidly evolving into not only a world-class institution, but the world-class institution addressing ocean science, ocean hypoxia, ocean acidification, ocean warming, all the things that we share a concern about. And there's the opportunity to find scientific ways to address them right here on the Central Oregon coast. I don't want to take too much time, but I do want to tell you during the last legislative session, we brought money home to address a lot of projects along the coast. They were really kind of focused on clean water and healthy living, water systems and storage in Waldport, connecting the Port of Toledo to the local sewer department, trying to get some money to start addressing the need for a new dam in Newport, an $80 million project replacing earthen dams that are not only among the most vulnerable in the state, but probably are the most vulnerable in the state. And then $16 million to help Otis in the fire zone replace wells and septic systems that were destroyed or damaged during the fires there a year ago. By the way, the fire zone in Otis, among the hardest hit areas in the state during our floods last weekend. And you all understand well the potential for landslides and other issues in an area that's been ravaged by forest fires. The final thing may be of interest to you, and that is that the legislature has just gone through the every 10-year process of redistricting to draw district lines so that each House district and each Senate district have equal numbers of people living them. The redistricting process is designed to address population changes. It's an inherently political process, often has the potential to be an inherently politically ugly process. I won't bore you with all the details except to say that the boundaries of my own legislative district are scheduled to change with the election next year. My district will be shifting out of Tillamook County, out of Yamhill County and out of Polk County, and instead will be growing north. So I will pick up the thriving community of Yachats in South Lincoln County and actually extend a little bit further down the coast all the way to the city limits of Florence. Very excited about that. My district will also be extending inland to Philomath and to western Lane County. That's a significant change. And for those of you that live just south of or in Yachats or just south of there, I hope that's a change you find constructive. I won't talk about campaigns tonight. I won't talk about elections, but I did want to talk about the boundaries of districts and the representation that you receive in Salem. So with that, I will conclude. I will wish you a wonderful seminar tonight. The first 8 have been fantastic. I've always enjoyed coming down and joining you. I guess the one benefit tonight is that you don't have to drive as far in what could be difficult weather, and that you can enjoy your favorite beverage, which we didn't always get as much of a chance to do at the symposium directly. But have a wonderful, educational, and exciting evening. Thanks so much for joining us again. Thanks for all you do every single day.
All right. Thanks so much, Representative. I'm really excited that we were able to welcome you this year with the Cape Perpetua area being for the first time in your district. And I always just felt just a little bit short. And so it's great to have you officially representing this area of the coastline as well. We're really excited for you, and I appreciate your welcoming remarks there. I just want to remind folks that we'll be hearing, our keynote speaker tonight, Mark Hixon, who I will introduce in just a moment. But we'll have 3 sort of rapid talks right after that, and we're not going to stop for questions along the way. We just want you guys to put those into the comments. So if everybody could put comments or questions that you have in the comment section, we will address that at the end when we bring all the panel participants back up for those questions. So without further ado, I guess I should probably introduce myself. I am Charlie Plive, and I'm the Oregon Policy Manager for Surfrider Foundation. And this is, again, like our 9th year doing this event. It's pretty amazing how far we've come. So, but I'm go ahead and introduce Mark. Mark Hixon's our first speaker. He's doing our keynote talk tonight, Coastal Connectivity. John Muir was right. So a little bit more about Mark. Mark is a professor in the University of Life Sciences at the University of Hawaii at Manoa. He is the Sidney and Erica Hsiao Endowed Chair in Marine Biology and serves as the chair of the Zoology Graduate Program. His expertise in marine ecology and conservation biology, especially regarding coral reefs, which he has studied in many locales around the world. His research addresses the questions of what determines the number of fish in the sea, how so many different species naturally coexist with each other, and how marine reserves and artificial reefs help to conserve sea life and enhance fisheries. In 2004, Mark was recognized by the Institute for Scientific Information Citation Index as the most cited scientific author on coral reef ecology in the United States. This year, he was recognized as a Scientist of the Year by the Arcs Foundation Honolulu chapter for his remarkable record of research and publication in the area of marine ecology and conservation biology. As a mentor to graduate students, and for his efforts to educate the public about threats to our coral reefs. Before heading to Hawaii, Mark spent a number of years here at Oregon State University and co-chaired the original community team for the Cape Perpetua Marine Reserve. So without further ado, I'd like to welcome Mark.
Thank you so much, Charlie. It's wonderful to be here this evening with all of you. Always nice to visit the Oregon coast, even virtually. So let me share screen here, get into the slides. So it's really an honor to be here this evening. I've been asked to review the concept of ecological connectivity in Oregon's ocean. So I'm not talking about Wi-Fi here. I'm actually talking about the movement of molecules and the movement of organisms that connect different locations on our globe. That's what connectivity is. And those connections create a web of life about which I want to introduce you. So you're going to see a lot of arrows in my slides. I think almost every slide has arrows in it. So I'm going to focus and organize my talk around two truths. The first truth is that all things are connected. This is a truth that Native peoples have known forever, and unfortunately, many people in modern societies have forgotten. And then, secondly, everything is changing, and everything is changing at an accelerating pace. What used to be no longer will be, and it behooves all of us not only to realize in our depths of our soul that all things are connected, which I believe is essential if our species is to survive, but also to understand the change that's forthcoming and prepare for it. Actually, the change that's already with us. So, number one, all things are connected. On the right side there, standing next to Republican President Teddy Roosevelt in 1903 in Yosemite Valley, is John Muir. John Muir was a naturalist and conservationist who was a um, controversial figure, to say the least. But one thing he got right was his understanding about connectivity. And he famously wrote in one of his books that when we try to pick out anything by itself, we find it hitched to everything else in the universe. And that's the essence here. No matter what we do, we are affecting things not only in our own community, our own state, our own nation, but around the globe because everything is so interconnected. So the connectivity ecologically starts with molecules. It starts with molecules that are moved by the winds of the earth, by the rivers and the flowing waters of the earth, and of course, by the currents within the ocean. And these flowing molecules move things. They move pollutants as well as nutrients. They move oxygen. They move carbon dioxide from burning fossil fuels. They move the little plastic particles Representative Gomberg spoke about. It's those physical processes that cause a lot of the movement in the world. Now, this is the Land-Sea Symposium, so I want to start with species that very, very realistically connect the land and the sea. And of course, sort of the paragon there are Pacific salmon. And if you don't know that cycle living in Oregon, then you clearly have been missing elementary school. So salmon, of course, spend their growing years in the ocean, feeding off the ocean's productivity, then eventually migrate into freshwater where they spawn and typically die. Their young then work their way back to the ocean and the cycle continues. Now, although everybody knows that, salmon do more than simply move fish into fresh water and back out to the ocean. They're also extremely important in fertilizing plants, often hundreds of miles inshore. So after the salmon dies and its body rots, or if the salmon is eaten, say, by a bear or another predator and defecated, those remains fertilize vegetation far, far from the ocean. So it's literally transferring nutrients from the ocean way inland and keeping our trees and other plants growing in a healthy way. All things are connected. Another land-sea connection in the Cape Perpetua area is the marbled murrelet, which is an amazing little bird that nests on the branches of moss-colored old-growth forests, often far from the sea. In fact, up to 50 miles inland, these birds will have their nests and then fly all the way to the ocean where they feed on and collect small fish that they return to their nests. A very remarkable feat for a bird. And of course, because old growth forests are endangered by and large, and the ocean productivity is altering, it's not surprising that the marbled murrelet is an endangered species. So that's land and sea connections. This is not my expertise. I'm a marine biologist. So let's go entirely into the oceans now, where I'll feel more comfortable. As we get close to the shore, the extremely important currents that occur are called upwelling and downwelling. If you look at the left-hand side, where we have typical spring and summer conditions off the Oregon coast, the interplay of winds and the Earth's rotation causes surface waters to move offshore and be replaced by deeper waters that come from the deep sea that are filled with nutrients. These nutrient-rich waters fertilize the coastal ecosystem, allowing the phytoplankton or single-celled plants to proliferate, which then feed small animals such as the zooplankton, and then small fish and larger fish and larger fish and larger fish, all the way up to seals and sea lions and sharks and human beings. The reverse of that tends to happen during the winter when cold fronts move through and downwelling occurs, which sort of shuts off the supply of nutrients. Plus, it's darker in the wintertime, so the productivity is typically not as high. And all that production at those low trophic levels, as they're called, here you can see the phytoplankton and the zooplankton down at the bottom here. That feeds an entire web of life in our oceans. So this diagram simply shows for part of Oregon's ocean, this is certainly not all the species involved, how larger and larger zooplankton feed on each other till you get up to the fish, then the fish feed on each other, and this doesn't even go to the top. Of course, you have the seals and sea lions, the sharks, and humans up there. So you've got this incredible web of life. Things are getting more and more complicated. I'm building on the layers here. But what makes it even more complicated is that every species has its own life history. And during that life history, it changes ecologically, both in terms of its size and in terms of where it lives and what it does. So a typical example are many species of rockfish off the coast of Oregon. This is a typical species, the boccaccio. The boccaccio adults live offshore in deeper waters. They eventually spawn and produce little small larvae that undergo the dispersal phase of the species, where these larvae can drift far and wide. I'll be spending quite a bit of time talking about that. Then the juvenile fish eventually reach inshore or drifting nursery habitats, especially eelgrass beds and kelp forests. And then as they grow, eating smaller prey and become larger and larger, they move offshore and join the adult population. So this kind of connectivity is incredibly important to know for managing our fisheries as well as conserving life in the sea. If we want to for example, to catch a lot of boccaccio rockfish in a fishery, we'd better make sure that the inshore nursery habitats are healthy so that these offshore populations become replenished. And if we want to know over how large an area we need to manage a stock of fish, that's a local population of fish, we need to know what larval dispersal is like. How far do the larvae disperse? Much connection is there after adults spawn to where the babies eventually settle out? Very important questions that science is only just now starting to reveal. So I want to spend a bit of time talking about larval dispersal. And the first part of that is that the larvae of ocean fishes and animals without backbones, the invertebrates shown here on the left, are very different typically from the adults. In fact, if you look at all these invertebrate larvae on the left, you probably aren't going to recognize anything. But over here, we have most of the sea stars and their relatives. Down here are the mollusks, such as snails and clams, and polychaete worms. The crabs and their relatives, including barnacles, are over here, corals, sponges, and some zooplankton that don't live close to shore typically. On the right are various types of fish larvae, some of them quite comical looking, but very few of which look like the adults because these are zooplanktonic organisms drifting and feebly swimming in the ocean. And they're quite small. You can see the little inset here, sorry about the dirty finger, of how small fish larvae actually are. So the key question about this larval dispersal is, where do the larvae go and where do they go where they're fairly abundant? This is extremely important in the context of marine reserves, such as the Cape Perpetua Marine Reserve. It's well known that if you set aside an area from fishing, that fish will become more abundant in that area and they'll grow larger and they'll produce what's just called the Boff effect. Big, old, fat, fertile female fish will produce lots and lots of larvae. But all that is fairly meaningless to fisheries unless those larvae replenish fish areas and allow fish populations to continue to be exploited. So, how can we possibly measure larval dispersal? These little dinky animals drifting away from where they're born in the ocean, sometimes short distances, sometimes hundreds of miles. Again, this is important in the context of marine reserves. We now have 5 small reserves along the Oregon coast, including Cape Perpetua. So, how to measure larval dispersal? This is going to be the most complex slide I show you today. The first approach is a modeling approach. That is, you're not really even working with real organisms. The oceanographers can go out and measure currents for a while and start creating mathematical models that sort of predict where currents go at different depths of the ocean, how fast the currents are, how much they form little gyres and spirals and eddies and things of that sort. And once they've set up a numerical model, they can release little particles in that model. Again, this is all mathematics. They call these virtual larvae, little passively drifting particles. So the map on the right is an accurate map of the Oregon coast, which corresponds to these black lines you see on each of these graphs. And these are model runs from where the ovals are, which is the location of the release, or if you will, where these virtual larvae are spawned. You can see some here near Newport, some further down near Hsiao Bank. Many people call it Hecate Bank. And the two graphs are just two different depths. Larvae released at 164 feet versus 66 feet. And if you look at the colored lines, it looks like spaghetti. And that's what it should look like because it shows how complex the patterns of dispersal are. The different colors represent different release dates. This is all modeled from 2001, work done by Jack Barth and his colleague at Oregon State University. So what do you get from all this? One is that larval dispersal is messy, number one. Secondly, you'll see that some larvae, after 30 days, these are all 30-day runs, which is sort of typical larvae, some of the larvae end up way offshore, where they will almost surely perish and not find the inshore habitats of the adults, whereas others stay close to shore. And it all depends upon when and where the larvae are released and ultimately how they behave. So the modeling approach sort of tells us the realm of what might be possible, but it's much more satisfying to actually bring in real organisms. And this is where the genetics approach comes in. By looking at the detailed genetics of individual organisms, we can sort of see where related organisms or animals hang out in the ocean relative to other groups. And this especially works with sessile organisms like barnacles, which once they settle, that's where they're going to live their entire life. So in this particular study, also along our coast, you can see what I showed in the previous diagrams where the offshore current sort of heads offshore at Hsiao Bank, which sort of directs the current offshore, creating a down current eddy near Hsiao Head. And what this genetic study showed was that because of this eddy that forms off Hsiao Head, the barnacles that live there are genetically related to each other and not very genetically related to barnacles either north or south of there, suggesting that larvae are retained within this gyre and there's not a lot of larval connectivity to other areas. But this is still fairly indirect. But fortunately, the newest methods are showing us that are showing us the real patterns of larval connectivity. And this is called genetic parentage analysis. And it's very similar to DNA fingerprinting that humans use when, say, you're having a paternity suit or something like that. With this method, very detailed genetic analyses, we can actually find parents and their offsprings in the ocean and identify them, which means absolutely you know that that particular small fish came from a parent elsewhere in the ocean. Now, to my knowledge, this has not been a completed analysis off Oregon yet. The closest analysis I found was conducted down in California, so I want to review that for you. It is with a species that occurs up in our area, kelp rockfish, shown there on the right. And this was a study done around Monterey Peninsula. So Monterey's here, Carmel's down here, Carmel Bay, the south end of Monterey Bay, 17-mile drive, if you know the Monterey area. So each one of these lines represents parent-offspring pairs. So the parent is the circle and the offspring is the triangle. So this shows that this fish that lives here in a small home range spawned a baby that went out in the ocean, didn't fly across the peninsula, drifted out in the ocean, eventually settled down here in Carmel Bay. And this is an exhaustive study. They sampled over 6,000 fish. And what's nice about this method is you don't have to kill the fish. All you need is a little tiny clip of their fins to get enough genetic material. But over those 6,000 fish that were sampled, they found, needle in a haystack, eight parent offspring pairs. And you can see that 3 of those parent offspring pairs were spawned off Monterey, and their babies now live in Carmel Bay, which shows that this marine reserve up here is replenishing populations in Carmel Bay. There was one example of the reverse pattern, which shows how complex larval dispersal is. That is, the parent is in Carmel, the baby lives up in Monterey. And then there's several cases of not much larval dispersal at all. That is local retention of the larvae, probably by some kind of local gyre or eddy. And one case of larval dispersal just within Carmel Bay. So this sort of information, which is unequivocal, tells us that local populations of rockfish are replenishing other populations of rockfish just about 15 miles away or so, at least these kelp forest species. Very important information for both managing fisheries and for conserving species in the ocean. Now, in the future, we can take this parentage analysis and do even more important things with it. And this is what we call fish flow. So this hasn't been done yet on the West Coast. This is a study my lab conducted on an easy-to-study species in the Bahamas, this damselfish. And the reason is to really understand fish flow, you need to measure egg production by the female. How many eggs are being produced that produce the larvae? And the nice thing about these fish is that they lay their eggs on the seafloor, and you can get them to do so in little tubes and count the number of eggs without having to kill fish. So on the left, you can see a map that's very similar to the last slide. This study also found 8 parent offspring pairs after sampling lots of adults and juveniles. Some of the larvae some of the babies were found right where their parents were. That's little circles here, and others moved between islands. Now, knowing population size and egg production, you can then calculate the actual proportion of larvae that survive larval dispersal. That is the actual flow of fish, not just that there is connection, but how strong the connection is. And in the lower map, you can see that Cat Island has very strong larval retention. So this is a population that keeps itself replenished and does some replenishing of other areas. So that kind of information is even more important. Now, if we combine that information of fish flow with what happens to the catch of fish from fisheries once it's brought to shore, then we can actually follow fish from spawning to supper, which is a remarkable feat that will soon be accomplished with ongoing studies. As sort of a mock-up prelude to that, this is another study my lab conducted, this time in Hawaii. This is along the west coast of the big island of Hawaii. So for scale, this is about 100 miles, which is about a third of the Oregon coast. And my study, which was led by Mark Christie, the other study led by Darren Johnson, both in my lab. They've gone on to become professors of their own. We did the ocean study where we looked at larval dispersal of a coral reef fish, a typical species, and we found that larval dispersal was primarily from south to north along this coast, including from marine reserves, replenishing areas outside the reserves. An independent study conducted about the same time by Jack Kittinger and his colleagues looked at reef fish landings at this particular port and where people took the catch after they caught it. So this is recreational fishing, people either giving fish to their friends and family or taking the fish home and eating them. And if we combine these two data sets, and if they are representative, they tell us a very important story. First, that if we're going to manage fisheries on the Big Island, we have to include the entire coast. But secondly, and I think very important from a conservation perspective, is that people eating fish on the north half of the Big Island had better be concerned about the status of coral reefs on the south half of the island, because that's where their fish come from, even though they catch them up here. That connectivity, knowledge of fish flow in detail, I believe is the next phase of having an engaged citizenry um, regarding fisheries in the ocean, as well as improved management of fisheries. Okay, I want to finish up, and this will just be a few slides on how everything is changing in the ocean. We have all these connections that we're learning about, very complex, and on top of everything else, everything is changing. Everything, everything, everything. We know that's true on land now with the droughts that have been happening and the wildfires and the floods in Oregon. Things are happening in the ocean as well. And here I'm referring to the evil twins of ocean warming and ocean acidification, which are both the result of human carbon emissions from burning fossil fuels and deforestation. Now, this is a complex graphic, so I just want to go through it step by step to get you introduced to the main changes that are occurring. First, with a warmer atmosphere, storms are increasing in frequency and intensity, and that's obviously going to affect ocean ecosystems very close to shore, not to mention coastal communities of humans. Associating that with sea level rise, we can see that estuaries and rocky intertidal systems are going to be shifting fairly dramatically, along with the loss of houses that are built too close to the beach. All the currents are shifting. This includes upwelling currents as well as major and minor currents close to shore or far from shore, El Nino, La Nino patterns. All these are shifting right now. Very difficult to predict exactly what the outcome will be, except that definitely the ocean will be getting warmer. And there's some ramifications now we'll see with upwelling. That excess carbon dioxide in the atmosphere, when it's dissolved in seawater, reacts with water molecules and forms an acid, carbonic acid, which is making the ocean less basic, or we call it more acidic, ocean acidification. This affects organisms that have calcium carbonate or limestone skeletons, and I'll give you some examples of that. And of course, all the species that interact with those species. And then finally, species shifts. As the oceans get warmer, we're seeing a wholesale shift of the geographic distribution of species toward the poles. Species basically seeking out the water temperatures they prefer and staying away from water temperatures that stress them. So let me give you quickly 4 examples of these issues. Trying to model exactly what's going to happen is very difficult. This was one approach for Columbia River Chinook salmon that tried to look at temperatures of the river, as well as different flows based on shifting rainfall patterns, temperatures behind the dams, temperatures in the ocean, along with upwelling. And even with all that being modeled, they left out ocean acidification. Now, an acidifying ocean doesn't really directly affect salmon much, as far as we know, but it very much affects what they eat. So single-celled plants called coccolithophores have calcium carbonate skeletons. Small planktonic snails called pteropods have little shells made out of calcium carbonate. And both these species or groups of species are being negatively affected by ocean acidification. Therefore, there's less food for baby salmon and other fish. Everything is connected. Dead zones are starting to intensify off our coast. That upwelling I spoke about earlier that brings nutrient-rich water into shallow areas to feed our phytoplankton is increasingly becoming less and less oxygenated as the oxygen minimum zone, a natural phenomenon, is moving shallower. So we have low oxygen in these upwelled waters, and when surface waters stagnate and the phytoplankton dies and sinks to the bottom and rots, that pulls even more oxygen out of the water, and we end up with hypoxic conditions that kill fish as well as things like Dungeness crab. This is a bunch of Dungeness crab that died. They weren't able to crawl out in time and washed up on the beach. This is being exacerbated by a warming ocean. Species shifts. Some species moving into our area may be great, like some of the larger tuna species. Others may not be so great. The Humboldt squid is a 6-foot long, highly voracious predator that's been moving into our area during El Ninos. Never get in the water with one of these things. They will attack you. And in Chile, where they've been moving to the south away from the equator, they've been implicated with the decline of hake populations, one of our greatest volume fisheries here off Oregon. And then finally, the warming ocean is resulting in either new microbes moving into our area or bacteria and viruses that were already there getting stimulated in stressed organisms. And one of these is sea star wasting syndrome, first observed off our coast in 2013. It's affected over 20 species of sea star, possibly caused by a virus, but especially affecting sunflower sea star that have declined by over 90%. This is a keystone species. These are all the different prey the sea star eats. You can imagine what it's like when the predator is removed. Predators are known to regulate the abundance of their prey, that is, to stabilize ecosystems. This species is now considered critically endangered. And as the authors cite, while it's unclear whether warming climate triggered the outbreak, it certainly is making it worse. So, last slide. All things are clearly connected and changing. I don't want to leave you depressed, though. There's things every one of us can do to address and adapt to these changes that are coming down the pike to us. So what can I do, or what can anyone do? It boils down to two things. Besides educating yourself and others, which you're all doing by being here, thank you. Number one, less pollution. We have to not consume so much. We are such a consumer society, generating all these plastic particles, using too much fertilizers on our lawns and golf courses, as well as agriculture, sewage systems near the coast, junk, garbage, pollution. The ocean's downstream from everything. We need to just stop dumping so much into the ocean, as well as stop emitting so much carbon dioxide that affects the ocean. And secondly, we need to implement more marine reserves than we have implemented everywhere in the world. It's been found that once an area is set aside from other human impacts, that area, both on land and in the sea, is more resistant to climate disruption, more resistant to an ocean warming, more resistant to an ocean acidification. It's the best thing we can do to help our marine ecosystems change more slowly as these inevitable changes come down the line to us. So thank you all so much for listening, and I'll be happy to answer questions when it comes time for the panel discussion. Thank you.
All right. Thanks so much, Mark. That was great. I want to remind folks to put your questions in the comment area. I saw a handful of those coming through, Mark, so I know that you have stimulated the interest of our audience. So now we're getting ready to head into intermission. So this is normally, if we were in person, the time when I would tell you where the bathrooms are and where to go get a beer and stuff like that. But we're going to leave you to yourselves for a few minutes. Also, though, we're going to have a video from the Oregon Coast Aquarium. So during the intermission, the aquarium will provide some insight on one of the best ways to connect with Oregon's coast, tide pooling. The Oregon Coast Aquarium understands the importance of fostering the connection between people and nature, and tide pooling is a great way to make those connections to some of Oregon's wild spaces. So go get a beer, go use the bathroom, tune in for a few minutes here and join us as we discuss the intertidal zone, its inhabitants, and tide pool etiquette in this short intermission video. And we will be back at 6:30 for our next presentation.
The Oregon Coast Aquarium understands the importance of fostering a connection between people and nature. A great way of feeling more connected to Oregon's beautiful coast is by taking a closer look at some of the animals that live there and exploring this unique environment. We're here to provide that closer look, foster connection, and inspire further exploration that can ultimately lead to stewardship. So the aquarium is home to an assortment of animal ambassadors. Animal ambassadors are representatives of their species who help educate the public about their wild counterparts. Ambassadors are a vital link in marine education and conservation, as they help visitors make connections with the representative species in the wild.
So here at the aquarium, we have many of the tide pool animals that you'd see if you're out tide pooling somewhere along the coast. And we like to talk about different ways to interact with the animal that's safe for both the guest and for the animal. So if you're out tide pooling, so I'll demonstrate some of that. But the aquarium is just a really great place to come and get a closer look at a lot of these animals that you might find if you're out there tide pooling on your own. Here in Oregon, we actually have 2 high tides and 2 low tides a day. So the intertidal zone is an area where the water is kind of moving back and forth. So it's a really tough place for lots of animals to live, because sometimes they're inundated with water, sometimes they're exposed. There's a lot of wave action. If you've gone out on a stormy day, you see all the waves crashing on the shore. All these animals have to deal with this really dynamic environment. The intertidal zone is actually broken up into different areas. I happen to have a nice diagram behind me that I can show you, but the highest area is called the splash zone. That's the area that doesn't get wet, inundated very often unless it's a really high tide or a storm. The animals that live up there are usually shells like barnacles or limpets, and they have a way to actually preserve water. They hold water in their shells. So it's usually not very diverse up there, but you see a lot of barnacles and those shelled animals living in the spray zone. The high tide zone is the area that gets covered during high tide, but it's exposed most of the day. So it'll be covered just for a little bit of the day. Those animals are able to be out of the water for a long time as well. But they also get inundated more often than the animals up in the spray zone. So you'll see barnacles there, you get into some mussels, and you might see some stars in that area as well. We have the mid-tide zone, which is kind of the most active area. So that's the area that's covered and uncovered every tide. So that's a great area for sea stars and anemones and a lot of other animals. It's nice because when the water comes in, it brings food and it cools them off. So it's a really active area, and you get a lot of life living right in that mid-tide zone. Then you have the low tide zone, which is most of the time is covered in water, but it's only uncovered on a really, really low tide. So most of these animals it gets a lot more diverse in that area because there's water pretty much all the time. So you get a lot of kelp, you get plants, you get different kind of chitons, and the subtidal zone is the one at the bottom, and you start to get wolf eels and fish and different animals too. Now we're talking about tide pools specifically, and tide pools are pretty cool here locally in Oregon. They form in areas where the rock has been eroded away, the rock along the coast, and then it leaves little nooks and crannies and holes behind. So whenever the water comes up and then it recedes back out into the ocean at low tide, there are these little pools that form. They can be deep, they can be really shallow. And there's lots of animals, they're like little oases where the animals will kind of hang out until the tide comes in again. It's a really neat place to go and look for different critters. And we have a lot of the animals here at the aquarium that I'll show you here shortly. But tide pools are really a difficult place to live because the conditions change really fast in them. So if you imagine a little puddle or a little pool of water on a sunny day, the water heats up really fast. So these animals have to adapt to a lot of sudden changes. So the water can heat up really fast. When the sun is beating down on the water, it evaporates and it leaves the salt behind. So the salinity, the salt levels increase a lot too. So they have to adapt to that. They're living in pools with their predators. So they need to be able to camouflage and defend themselves. A lot of animals have a way to defend themselves. And also the oxygen levels decrease over time when they're in these little pools of water. So it's a really kind of stressful environment for a lot of animals to live. So the ones that live there have to be really tough and adaptable and have a lot of really cool adaptations in order to survive there. So when you come to the aquarium, we have a lot of those same tide pool animals you'd see when you're out tide pooling out along the coast here. But this is a safe environment for them, and you'll notice them doing different behaviors that you wouldn't see them necessarily doing when they're out in that more harsh tide pool environment. So you might see a barnacle feeding, for example. We do like to talk about etiquette and how to properly interact with the animals in a way that's safe for both the animal and for you. So I'm going to demonstrate a couple techniques and show you a couple different animals and the best way to interact with them if you're out at a tide pool. Anemones are pretty commonly seen out at the tide pools, but these are two of the more common ones. We have a giant green anemone here. This is a fish-eating anemone, which is actually really cool. And they're called cnidarians. It's a group that they're in. So they're actually cousins of jellies and corals, and they hunt or catch their food by stinging. So they have tentacles similar to jellies, and their tentacles have little stinging cells called nematocysts. So if I'm going to reach in there and touch them, and when we do, you just use one or two fingers really gently. We call it creature reachers. You can reach in there, just really gently touch the anemone. So I'm going to touch it. And you see it's closing up. So it's kind of probably kind of a defensive mechanism, or they just kind of fire off their little stinging cells in case it's something they might want to eat. So I'm not something they want to eat, but you see how it's kind of sticking to me. Their little stinging cells are kind of like harpoons. So if I was a fish or a really small animal, it would just hold on to them, stun them to the point where they could pull them into their mouth, which is that hole in the middle, and then start to digest them. And most people don't react. Some people, if you react at all to the stinging cells, your fingers might go numb, but that's it. They're not dangerous enough. Our fingers are too thick for it to actually really hurt us. The next animals we're going to talk about are sea urchins, and you often see these out at tide pools, and they're a popular animal to come and look at here at the aquarium as well. And these animals are called echinoderms, and they're actually related to like sand dollars and sea stars and sea cucumbers. But the coolest thing and most noticeable about the urchins are their spines. So that's a way for them to defend themselves. And it's also a way for them, they'll often jam themselves into these like rocky crevices. They can spread their spines out and kind of press against it. So when the waves are crashing in, that's a way that they hold tight. Another cool thing that they have are tube feet. They're like little, looks like little tentacles almost sticking out with suction cups at the end. They don't have stinging cells like the anemones do, but they just have, that's how they hold on. They'll grab food that way, and they can pass it along to their mouth, which is actually underneath their body. And it's also, they also will sometimes camouflage. If they think there's a predator or something, they'll grab a shell or a piece of plant and they'll hold it on top of themselves to kind of hide if they're in an area that might be a predator. The best way to interact with them is carefully. You notice that they're very pointy. They have those spines, and the spines can actually, if you were to step on one, could break off into your skin, so it wouldn't feel very good. So if you see them, the best way, I usually use one finger. I like to reach my finger in kind of in between the spines, and you'll notice all the spines will kind of hug my finger. These guys primarily eat plants, so they'll eat kelp, and they have really interesting mouths. They're called Aristotle's lantern. It looks like 5 little plates, kind of almost like a beak, and they'll scrape algae and different things off of rocks. The barnacle is one animal I really like to talk to guests about because a lot of people don't even recognize it as an animal at all when they're out at the tide pool. So you'll often have to kind of walk past them to get to the tide pools because it is usually up in the area where it's out of the water. And when they're closed up in their shell, they basically just look like rocks. So I've gone out to tide pools and seen people just walking all over the barnacles. So it's really neat to be able to show people what a barnacle looks like when it's actively feeding and opened up in the habitat here at the aquarium. They're actually crustaceans, so they're related to lobsters, believe it or not. And when they're born, they swim around their little larval stage. They don't have a shell. And basically what they're looking for is an area where there's going to be a lot of food. When they find that, they'll actually glue their head. So they have a really strong cement. They glue their head to whatever rock or wherever they want to live in a place where they think there's going to be a lot of food. Some barnacles, species actually grow on whales, and the whales will take them to the food. That's a pretty clever strategy. But when they glue their head, they'll actually grow a calcium shell around them, and then their legs will come out. They look like little eyelashes. They're called cirri, and they're filter feeders. They'll come out and they'll start grabbing food as it floats by. So it's really fun when you get to watch one eat. But when you see them at the tide pools, they're just closed up holding the water in, so you usually don't get to see the body coming out. And probably the most iconic animal that you can find in tide pools, especially out here in Oregon, are sea stars. There's lots of different species of sea stars. The most common one that we see are ochre stars. And they can be out of the water the longest of any sea star. They can be out of the water for about 50 hours. They're actually able to hold in water. And they come in different colors. I see purple ones, orange ones, kind of yellow ones. They're related to animals like sea urchins and sea cucumbers and sand dollars. They're kind of like the wolves of the tide pool. So for an invertebrate animal, they're pretty fast. So they move faster than the other animals. So they can run them down. And they have different ways to actually pry open the food. So some, like the ochre star, will actually evert its stomach out, kind of inside out, and it can start to digest whatever it's trying to eat before it actually pulls it into its system. They'll eat things like snails and limpets and they're mainly carnivores. So they eat a lot of those little animals that share the tide pools with them. So ochre stars generally have 5 arms, but I think sunflower stars can have up to 24. So depending on the species, they have different amounts of arms. And they actually move really well with their tube feet. So they have hundreds of little tube feet that run along each arm. And that's what they use to kind of crawl along the seafloor. They hold on really tightly to the rocks. So all the animals you see at the tide pool, it's best not to try to pick them up because you can actually damage them if you're trying to like pull them up off of the rock. So always good to look at from afar. You can gently touch them with 1 or 2 fingers, but best not to actually try to pull up on the sea stars.
Adventure awaits for those who have been inspired during their visit at the aquarium to continue their exploration of wild places all along the Oregon coast. The best place to view the intertidal animals we met at the aquarium are at local tide pools. A few tide pooling locations close to the aquarium include Yaquina Head Outstanding Natural Area, Seal Rock State Park, and Cape Perpetua's Marine Reserve. Before you go, there's a few things you must know. First, get informed. Know the rules of local, state, and federal parks. Then, check the tides. Make sure you plan your visit at a low tide when the tide pools are visible. Lastly, make sure you wear appropriate clothing for the weather and terrain. Sturdy footwear is highly recommended. When you get to the tide pool, it is important to stay safe. Always keep an eye on the ocean and be on the lookout for sneaker waves. Also, be careful. Tide pooling areas are wet and slippery. As you explore the tide pools, it is important to be a respectful visitor to this fragile ecosystem. Tread lightly around the pools and watch your step. Make sure each step is on solid rock. Avoid stepping on any animals attached to the rocks around the pools. You are welcome to touch the animals that you see with a gentle 2-finger touch, as you saw Jeff do with our intertidal animal ambassadors. Just please don't remove them from where they are attached. Lastly, while visiting the tide pool areas, you might be fortunate enough to see other marine wildlife, including shorebirds and marine mammals like harbor seals. These animals are protected under federal law. Please give them plenty of room by keeping a safe distance away. There are several ways you can show your appreciation for marine environments, even when you are out visiting tide pool areas. One of the easiest ways to give back is to pick up any trash you find during your exploration. You can also support the protection of wild places in future legislation, and even help scientists collect data about marine environments by participating in citizen science projects. The most convenient way to tap into opportunities with citizen science is to download the app iNaturalist on your mobile device, or by visiting www.inaturalist.org. Thanks for joining us and taking a closer look at some of the amazing animals living here at the aquarium and exploring opportunities to safely visit their wild counterparts at local tide pools. We hope to see you at the aquarium soon.
Awesome. Well, thanks so much to the folks at the Oregon Coast Aquarium who put that together for us. Tide pooling is something near and dear to my heart. And for Cape Perpetua, I should say that it has one of the longest, most continuous intertidal and biodiverse intertidal zones in North America. So a big shout out to like our folks in the Cape Perpetua area who were involved in the ambassador program, doing outreach in the tide pools throughout the summer. And I would encourage anybody to check out the tide pools in any of our marine reserves. They're all great places to tide pool. So now we're going to transition into our lightning talks. We're going to be going pretty quickly here. Each speaker is going to have about 10 minutes, and so we'll be transitioning pretty quick. So keep your questions coming in the comment section. We're going to start with Julie Gonzalez, who's a Ph.D. candidate at University of California, Davis, and is a current NOAA Margaret A. Davidson Fellow with the San Francisco Bay National Estuarine Research Reserve. Julie is interested in estuarine community ecology and how to use this science to improve coastal habitat restoration while facilitating collaborative co-development of research projects and considering multiple stakeholder perceptions in the process. Over the past 8 years, she has been involved in several restoration projects along the California and Oregon coast as both a research scientist and a former project manager with California State Coastal Conservancy. So I'd like to welcome Julie, who's going to give us a talk here on determining salt marsh restoration outcomes using focus groups in ecological data. Take it away, Julie.
Thanks. Okay, that's yes.
So Julie, can we just have you maybe turn your camera off? Because I think the feed is just a little slow.
Got it. That better?
Nope, it's actually your audio feed that we're getting some feedback on. If you're using wireless headphones, if you can switch to wired or maybe none.
Okay, how's that?
We've still got the feedback. It wouldn't be a virtual event if there were no tech issues. No, it's not better yet. If you have wired headphones, you can try. We can actually hop to our next speaker and come back to you.
Let's see.
Okay, Scott, we're going to move to you. Charlie, I'm bringing you back up, and then we'll give Julie another try here in a minute.
All right, I'm going to introduce our next speaker, who is not Julie for the moment while we work that out. Another individual who's been deeply involved actually in the marine reserve process over the years in Oregon, Scott Heppell, Associate Professor of Marine and Freshwater Fisheries Ecology at Oregon State University. Scott's research interests are the physiological ecology of fishes, in particular how physiology, behavior, and life history traits affect the interactions between fish populations, their respective fisheries, and the environment. He's worked on bluefin tuna on the Atlantic high seas, Mediterranean and east coast of the United States, groupers throughout the Southeast Atlantic, Caribbean, and Gulf of Mexico, and on rockfish in Oregon and Alaska, and on trout, steelhead, and salmon in Japan and in the high deserts of eastern Oregon and northern Nevada. He collaborates with academic scientists, state and federal agencies, foreign agencies, and universities and commercial and recreational fishermen working together to try and address issues related to the sustainability of marine and freshwater resources and their ecosystems. At Oregon State University, he teaches classes in fish physiology, fishery biology, and management of salmon in the Pacific Northwest. And like a couple of our other presenters tonight, Scott was also on the state's founding community team for the Cape Falcon Marine Reserve up on the North Coast. So with that, I will welcome Scott.
Great. Thanks, Charlie. Does my audio sound like it's coming through all right?
You sound great.
Excellent. Well, we'll go with that then. Very good. Thanks, Charlie, for that introduction. Thanks, Bree, Tara, Joe, and Paul for putting all this together and giving me the opportunity to talk today. I'm going to talk about the role of Oregon estuaries and recruitment early life stages of rockfishes, and I'm going to talk a little bit about fishes in general. Estuaries are really important, of course, and to come back to what Mark was talking about earlier, they really are that nexus between the land and the sea. And as it turns out, and as Mark mentioned, nursery habitats are really incredibly important. And I'm going to talk a little bit about that story as it relates to estuaries today. As I get started, I do want to acknowledge that a bunch of the work that I'm going to talk about today has been done by my graduate students. In fact, enough of it's been done that I've added Brittany Schwarzkopf as a co-author or co-presenter for this talk. But Brett Gallagher, Allie Dobble, and Amy Jo Lindsley also have done a lot of this work, and to them I am very grateful. All right, so we're going to start off some basics and a little sneak peek of what we've learned. So I'll tell you up front what I'm going to tell you at the end. The first, habitat is really important for fish, right? Doesn't take a lot of science to know that, but it's important. We're going to lay that out right up front. But here are the really important things that we've learned as we've done our work in Oregon estuaries over the year. One, natural habitats function differently than man-made habitats. As Mark alluded to, and in fact stated directly, things are changing a lot, and we're seeing that both on local and regional and global scales. Locally, we're seeing a lot because of development, because of pollution, because of changing water temperatures, changing acidities. Some of those are driven by local processes, some of those are driven globally. But it turns out that a lot of those local processes, particular development, where we're putting in pilings and docks and jetties and other sorts of development, creates habitat that fish can kind of hang out on, but it's very different than the natural habitats they're used to. The other thing I want to point out, and this is a good part of the story, is that the rockfish community in Oregon estuaries is actually diversifying. We're seeing more species in estuaries now than when we started this work 15 or 20 years ago. And the downside, coming back to that, is that natural habitats, particularly seagrasses, which is what I'll focus on today, are declining due to those local and global events, which may impact rockfish diversity and abundance. In fact, the diversity and abundance of all sorts of different species. So we're seeing good response that they're becoming more diverse, the communities are of rockfish in our estuaries, but habitat decline is a particular concern. All right. Mark mentioned essential fish habitat and nursery habitat. There is, in fact, a legal definition, according to U.S. law, of essential fish habitat. And it's, first of all, important, as we mentioned, EFH, as we're talking about here, is critical for conservation and management of all the things that we care about. And essential fish habitat, as defined legally, are those waters and substrates necessary to fish for spawning, that is making new babies, breeding, part of that process as well, feeding, and growth to maturity. That's the legal definition of essential fish habitat. We don't necessarily have a legal definition of nursery habitat, but we have some operational definitions. And it is those habitats where juveniles rear and grow and contribute to the adult population. But it's important to point out that not all nurseries are the same. For example, and then this, what I have on the slide here with juvenile habitat number 1, maybe it has a lot more juveniles in it than habitat number 2. But what we really care about is not if there's lots of juveniles or a few, but how many of those grow and survive and make it to the adult population. So if only 25% of them are surviving, sure, it's juvenile nursery habitat, but maybe it's not that important. But if like 100% of them, which is, I suppose, a little optimistic, survive to adulthood, then maybe that's a much more important habitat that we might want to be concerned about. But essential fish habitat and nursery habitat generally are part of that connectivity cycle that Mark was talking about, because these things happen in different places. Okay. So some of the research questions that we've been asking in my laboratory with my graduate students and with others are, how do different habitat types affect fish and fish communities in Oregon estuaries? So graphically displayed here on the left is some piling habitat for a dock. Those happen to be concrete pilings, but we see them made out of wood as well. And on the right is a very traditional Zostera type seagrass bed, maybe a Zostera marina, Zostera japonica, but in any case, a natural habitat, very common, or at least historically very common, to Oregon estuaries. And so we ask a bunch of questions about, how do they differ between these? How do fish perform differently in these two different habitats? And so a lot of the work that we do is really centered around a very simple technique, not all of it, but basically we go out and we catch a bunch of fish to really understand, are these communities, that is all of the individuals of all of the different species, not just rockfish, how are they different in natural habitats versus these artificial habitats, docks and pilings? So we go out and we catch a bunch of fish, and we've been doing this for the last 18 years almost now, where we set these traps, pictured down here in the lower left. We call them minnow traps. You can order them online. And we set them for basically a 24-hour period, which samples through 2 tidal cycles. And we set them in both seagrass habitats and in artificial man-made habitats, docks and pilings. We do it during the spring and summer. We do it during the high tides. We do it during the spring and summer because that's when most of the baby fish are recruiting into estuaries and growing during the first year of life. We've been doing this, as I said, for about the last 18 years. We don't do it every year, but we've done it for a series of years. And we basically do very simple stuff. We collect, we measure, we identify, we count. And we look at each fish, whether it's a gunnel or a sculpin or a stickleback or a rockfish or an English sole or a lingcod, and so on and so forth. And then once we describe the overall community, we really have been interested in the last few years in focusing on rockfish and what they do. And so we retain some of the rockfish for genetic identification, do diet studies and growth studies to really look at, are there performance differences between these two habitats, which would indicate that maybe one habitat is better than the other. And the reason we use genetic identification is because when rockfish are small, sometimes they're pretty hard to tell apart. And so we've collaborated with the National Marine Fisheries Service lab in Santa Cruz and Carlos Garza's lab to do a lot of the genetic ID work, which has really helped us with our ability to understand our overall projects. All right, so we count, we measure, so on and so forth. And then we look, and we're really interested in, how do these communities change over time? And so what I'm going to show you next is a slightly complicated graph, which I think I can explain fairly simply. But you're looking at 2 different directional arrows here, or points in time. On the left, in the dashed line, is the community structure and how it's changed over time for dock and piling habitats. And on the right, with the solid lines, is how it's changed in seagrass habitats. Now, what each one of these points means is that we take the entire community, the abundance and diversity of fishes when we've sampled in a given year, and we assign it an individual value. So it represents a point. So for example, down here on the very bottom, 2008, that point for dock piling habitat, that point represents basically a single value assigned to the diversity of species in the system. And as you'll see, these walk through different locations over time. Points close together indicate that the communities in one year was very similar to the community in another year, and points far apart mean they're much more different. And similarly, if 2 points are close together between a dock habitat and a seagrass habitat, that means they're very similar. And if they're far apart, that means they're very different. And so we can see, for example, between 2008 and 2009, there was a pretty big change in the community around docks and a very small change in the community around seagrasses. And between 2009 and 2012, again, we see a bigger change between dock habitat relative to seagrass, the same in 2013 and 14, maybe similar changes in 2016, and again, bigger changes in dock habitats. So, and the other thing to note is that in 2008, very early on here, these two dots between dock pond and seagrass habitats were fairly similar. So that is 13 years ago, the fish communities in the two places were fairly similar. But by the time we got to 2017, they had really spread apart. They're very different. That the seagrasses are supporting a very different community of fish than the dock and piling. And in fact, the other thing to point out is that the relative amount of change between 2008 and 2017 is quite small relative to the total amount of change between 2008 and 2017 in the dock habitats. And as most of you, if you've lived on the Oregon coast for a while, know that there were some pretty substantial environmental changes that happened between 2008 and 2017. And in spite of that, we noticed that the community structure varied between habitats. It varied over time, but there's a lot more stability in the seagrass habitat over that time than we see in the dock habitat. So seagrass tends to be more stable, and while we can't directly relate that to being resilient, it might indicate that that's the case. And in fact, I was inspired enough, and I'm not a very good poet, but I wrote a poem inspired by Robert Frost about these two paths that diverge in Yaquina Bay. By time or hand, I cannot say. For one path indeed lay grassy fair, the other trod by human air. And be one traveler, long I stood, and hoped if nature restored it would. And that's really what I want to talk about, is the value of these natural habitats to our fish communities. So we know that they've changed as the overall communities, and now I'm going to narrow down a little bit and talk about rockfish specifically, because those communities were all of the different kinds of fish that were in the estuary. But what about rockfish? Well, if anybody's been paying attention, we know that starting in the late 1990s, and early 2000s, there was a groundfish crisis. Lots of rockfish, as shown here on the left, populations declining, being declared overfished. In fact, the vast majority of them were overfished. And of those species that are listed there, widow, canary, boccaccio, dark blotch, Pacific Ocean perch, and cowcod, since 2011, most of them have been declared recovered. And so our sampling of these estuaries spans almost the time from when they were declared overfished to the time that many of them were declared recovered, and generally our management of rockfish has been improving. When we started this work back in 2004, 2005, and that's Brett Gallagher's work, it didn't matter where he sampled. The only two species of rockfish he ever found were black rockfish and yellowtail rockfish.
Scott will need to wrap up soon.
Okay. When Allie Dobble did her work in 2012, she found 3 species. And when Amy Jo Lindsley did her work in 2013, she found 8. And now we are seeing 10 species of rockfish recruiting into our estuaries. And so that's really an amazing thing. And so just to wrap up, one of the things that we're seeing is that the seagrass habitats support better communities. We see some of these species only appearing in rockfish, or some of these rockfish only appearing in the seagrass. And I won't have time to share it with you tonight, but the last thing I want to throw out there is that we see that our rockfish in seagrass communities have more diverse diets and they grow faster. That's all great stuff. Our concern right now, and things to think for the future, is that we are also seeing declines in our seagrass communities. So while our rockfish are doing better, we are a bit concerned about the nursery grounds and how they might be declining. All right, I will stop there and leave it there. We can move on. Hopefully audio has been worked out for the other stream, and we can get on to our next talk. Thanks a lot.
Thank you so much, Scott. Remind folks to put your questions in the comment section, and we're going to quickly try and transition back to Julie, who's doing a talk on determining salt marsh restoration outcomes. So hopefully, Julie, you're coming through better now. Can we get you to test your audio?
Can you hear me now?
Oh, you sound fantastic.
Oh,
I'm going to pop
Excellent.
out here and let you take over.
Okay, thanks so much, Charlie. So my name is Julie Gonzalez. I'm with the University of California, Davis, and I'm here today to talk a little bit about our group's work trying to understand salt marsh restoration outcomes using focus groups and ecological data. And I want to start off by highlighting our end user partners who were absolutely integral in getting this project off the ground. The first is Paul Engelmeyer, who you were introduced to at the beginning of this symposium. Also, Sean Schuler and Saber Comet with the South Slough National Estuarine Research Reserve down in Coos Bay. And our project team is composed of scientists from UC Davis and Portland State University. Our project lead, Kat Deriver, is from Portland State, as is our social science technical lead, Melissa Hafner. So tidal marsh ecosystems are widely recognized as providing many valuable ecosystem services. They provide habitat for fish, as we just heard, crabs and birds. And also they can enhance water quality, they can store carbon, and also they're important for buffering storms and wave action. So 85% of vegetated tidal wetlands have been lost from West Coast estuaries due to diking, draining, and or otherwise converting these habitats. In order to combat loss of habitat and associated ecological and system functions, there's widespread marsh restoration occurring more and more frequently. However, there's often a disconnect between scientists and other user groups like community members and landowners about what successful restoration actually looks like. And this can decrease community support for restoration, which is often necessary to fund these projects. So our team asked the question, what does successful habitat restoration look like to different user groups? Our study took place at 3 bays along the Oregon coast, at Yaquina Bay, Alsea Bay, and Coos Bay. And we focused on a total of 9 habitat restoration projects across these 3 bays. This picture is one of those restoration areas within Alsea Bay. You can see the channels running through the restored area on the right here, which is demarcated by this line running through the middle of it and separating it from this unrestored area that's mostly green. And this picture is actually a really good example of what I'm talking about, that disconnect between public end users and scientists, because restoration can look unsightly at first when it's first occurring. And initially you might think that this right side looks worse because it's all brown. But even though this site is in early stages of restoration, it already has useful functions. And I'm talking about this sinuosity of the channels within this area that slows erosion and creates better habitat for fish and birds. So we gathered social and ecological data from each of these 3 bays to look at how perceptions of restoration outcomes vary across technical and public end users of these projects. Social data was gathered using focus groups that were made up of participants from 3 different user groups. The first was end users, so that's restoration practitioners and people directly involved in restoration projects. Next, we have direct information users, so folks who may not have been directly involved in the restoration, but people like scientists who have access to technical reports about the restoration. And finally, we have indirect information users. So that's folks like landowners and community members that are not involved in the restoration and don't necessarily read the technical reports, but are interested and may be affected by the projects. And we gathered 2 types of information from focus groups. Qualitative information, where we have conversations and highlight common conversation topics and key themes, and also quantitative information, where we get a numerical ranking of what folks value about restoration projects. And we also gathered ecological data by compiling monitoring data for 9 restoration projects across these 3 bays. And these were data related to plant cover, fish and wildlife use, things like water quality. We also went out and measured plants on the marsh to compare to previously collected monitoring data. And lastly, we'll have conversations with restoration practitioners involved in each project to put these data into context. So this project is still in early stages, but the focus groups were completed in August, and we wanted to share some of the preliminary results with you all. So the image here shows results from the quantitative portion of the focus groups that I mentioned, in which each participant is given a set of statements, and we ask them to rank those statements by which they value most and which they value less. So the top 5 values with which folks agree are shown in the red box up here on the right. And I know it's kind of hard to see this, so I wrote out these top 5 values on the left, and they are enhancing or increasing habitat for fish and wildlife, increasing ecological function in general, enhancing water quality, reducing the amount of pollution in water bodies, and also reconnecting tidal channels that were previously cut off. We also found that values were similar actually across the 3 user groups and across these 3 Oregon bays. And we were actually really encouraged and excited to see that across groups, people consistently value the same aspects of restoration projects. And this was supported by conversations in the focus groups where individuals from different user groups agreed about contentious topics. So this commonality was a surprising yet prevalent theme in these focus groups. However, something that was highlighted in these focus groups was the mismatch in scale between the metrics that are monitored at specific restoration sites, like plant cover, and the social values that tend to be at the landscape level, like increasing ecological function in general, for example. And no one project is going to be able to fulfill these values given the amount of degradation of estuarine systems. So in order to achieve these social metrics, it's going to take multiple projects and planning over time, and we need to develop metrics to measure outcomes on that larger scale. So for future work, we're going to finish gathering the ecological data and run analyses to look at things like percent change in plant cover over time and fish and wildlife use of habitat post-restoration. Also, now that we have key themes from the social data, we can go through the scientific reports for each project and calculate how frequently those values are identified by scientists in their technical reports. We will also compare assessments of project success using ecological metrics, measured and also social values, and look at how similar and different they are. So, for example, a project might be successful relative to its project goals as outlined by scientists, but these goals may not align with the values of the public end users, or they may align for some restoration projects, but not others. So we're really interested in looking at some of these nuances. Also, we are planning a future iteration of the project where we scale up to the whole West Coast and use surveys to gather social data instead of focus groups. And we hope to explore some of the same questions, but also explore this disconnect between landscape scale values and site-specific ecological metrics. And if we're funded for this, we hope you'll be involved in these surveys. So keep an eye out for that. So we hope that this work improves the assessment of habitat restoration by including more diverse community member perspectives. We also noticed that Oregonians from across user groups are thinking about success as how the estuary functions throughout the landscape. And this kind of goes back to this idea of connectivity that was talked about in the keynote. This work will help to address the mismatch in interpreting restoration success at the landscape scale and measuring it at the site scale. And this will hopefully motivate the connection of discrete restoration projects to facilitate restoration at that landscape scale. So some organizations and restoration programs, including the South Slough National Estuarine Research Reserve, are shifting to this idea of restoring from the headwaters to the ocean and focusing on how all of these habitats connect and how to restore landscape scale functionality to systems. Also, this is very important for organisms that use multiple habitats, like salmon, as Mark mentioned earlier, that use the estuary, the ocean, and the river networks. So lastly, we hope that our work helps to improve communication about this important work and increase public support for restoration projects. And with that, I will say thanks and looking forward to taking questions in the panel.
All right, thanks so much, Julie, for that. And yes, just a reminder for folks to go ahead and stick your questions in the comments and keep them coming. Just sort of to speak a little bit to Julie's talk there, this event some 12 or 13 years ago, the genesis of this really was an Ocean Issues Forum put on by the Watershed Council, the Scioto Watershed Council, really recognizing that connectivity between our watersheds and our oceans. So it's pretty cool to see this come full circle. Speaking of first full circle, it's really cool to introduce our next speaker, mariner and fisherman perspectives on ocean changes from Al Pazar. Al was the first person I rolled out the Cape Perpetua Marine Reserve proposal in front of that actively fished in the area. He's a longtime commercial fisherman residing in Florence, Oregon. He began fishing in the Pacific Ocean off the Washington coast with his father in 1964 out of the small fishing towns of Iwako and Westport. The 2022 season will mark Al's 58th consecutive year on the ocean. Al briefly attended Oregon State University, where he studied fishery science and met his wife, Pam. Al and Pam worked as a team together for 10 years on a small 36-foot fishing vessel. They've been married for 45 years and have 2 children and 1 granddaughter. Crab, salmon, tuna, and halibut were Al's target species for many years. Al and Pam have owned the Crab Kettle Seafood Market in Florence for 32 years and have provided Central Coast locals and visitors with fresh, right off the boat seafood since 1986. And in 2005, Al opened Local Ocean Seafoods on the Newport Bayfront with partner Laura Anderson, which I'm sure many of you guys have been to before. Al has always been interested in marine science, and in 2011, he purchased the 56-foot Pacific Surveyor to be used exclusively as an ocean-going research vessel, providing research scientists with access to the sea to perform their at-sea projects. Al and the crew of the Pacific Surveyor have worked with federal and state agencies and many educational institutions performing fishery stock assessment, critical habitat classification, water profiling, ROV, video, and acoustic surveys along the Washington, Oregon, and California coasts. Al served for 14 years on the Oregon Dungeness Crab Commission and was an inaugural member of the Oregon Council on Ocean Acidification and Hypoxia. And along with Mark Hixon, he co-chaired the Cape Perpetua Marine Reserve community team in its early establishment of the Marine Reserve. So Al was generous enough, he's out at sea right now, and he was generous enough to pre-record something, so he won't be available for the panel discussion, but we're gonna go ahead and parlay to his video now.
I want to thank Charlie and the organizers and sponsors of this Land-Sea conference for inviting me to speak to you today. And thanks to all you virtual attendees for showing interest in our coastal ecosystem, and in particular, the Cape Perpetua Marine Reserve and our associated marine protected areas. The video you are watching was taken by Lincoln City photographer Matt Brandt. More on that later. When asked to speak at this conference, I was asked to talk about my experience since the Oregon Marine Reserve program began in 2012, including ocean observations, changing ocean conditions and chemistry, and my experience with Oregon's Marine Reserve program. The theme of this year's event is connectivity. In the case of marine reserves, connectivity means different things to different groups of people. To those that study coastal ecosystems, connectivity might mean the east-west connection of marine life, rivers and creeks, old-growth forests, and marble muralettes, the ridgetop to reef ecosystem philosophy. A marine biologist might think of connectivity in terms of how a network of marine reserves might promote a safer environment for larval dispersal to occur and a place for juvenile rockfish to grow in size and numbers. The commercial fisherman might see it as unconnectivity. What once was a favorite area that might have been very productive fishing grounds are now off limits and present borders that now host the migration that comes with effort shift, the natural crowding in fishing that occurs when real estate is restricted and effort is not. A recreational fisherman may have to travel farther from port or be forced to fish in more crowded fleets in order to avoid restrictions around marine reserves. This is especially true of Cape Falcon and Cascade Head marine reserves. A concerned citizen or citizen scientist's connection might be the satisfaction that a certain amount of coastal state waters and their associated underwater habitat has been set aside for protection, much like state or national parks. He or she might volunteer their time to help in the research, monitoring, or enforcement of a marine reserve and the adjacent marine protected area. My personal connectivity surrounding the Cape Perpetua area is based on decades of almost daily interaction from the headwaters of the creeks to the western limits of the reserve. Living in Florence and basing most of my commercial fishing out of Newport has me driving 101, often almost daily. Much of my recreation has been fishing for steelhead in Cape Creek, Big Creek, or 10 Mile Creek. I have dug razor clams in 10 different locations between Berry Creek and Cape Perpetua. I have hunted rough grouse in the fall. For decades, I've laid hundreds of strings of commercial crab pots, transiting countless times over the now marine reserve location. When I trolled for Chinook salmon, the tack off 10 Mile Creek used to be one of my favorites. I cut my firewood, picked my berries off the logging roads between Florence and Yachats, and found Japanese balls at Tokati. Connectivity is in the human eye of the human beholder. I freely admit that most of my activities were extractive, taking something out of the ecosystem, although I'm pleased to announce that the only 2 green flashes I have witnessed at sunset were both off Highway 101 between 10 Mile and Cape Perpetua. I was always comforted that the extractions I did make were taken from well-managed and sustainable resources, from the crab and fish in the ocean to the firewood on a Forest Service permit. When then Governor Kulongoski made his executive order mandating more than 3 and less than 10 marine reserves to be established in Oregon's coastal waters, many groups and individuals made proposals for individual or networks of marine reserves. Some of these proposals were seen as overreaching and draconian by many stakeholders. At the time I got involved with the Cape Perpetua Marine Reserve Community Team, it was strictly for defensive purposes. I wanted as few marine reserves as possible in order to have our fishing lives disrupted minimally. The fishing industry was rightfully concerned. Dealing only with Cape Perpetua allowed me to focus and participate in what was truly a remarkable experience. To be a part of a large group with such diverse opinions, goals, and needs, taking a raw idea to a finished product, seeing the evolution along the 2-year exercise to arrive at a common and agreeable conclusion was amazing. I believe the marine science and biology components of the reserve issue were never really in question. It was an exercise in socio-economic community collaboration and compromise. So how are the marine reserves doing? I'm not a scientist, but I play one when I'm at sea these days. I have been fortunate in my research vessel business to get a good look at marine reserves and their associated comparison areas with some of the best high-definition cameras and lighting available. Last year, we performed a month-long ROV survey in California, looking at several hundred miles of coastline for their 10-year marine reserve review. We looked at dozens of spots from the Oregon-California border to Morro Bay. I was astounded at the amount of and variety of fish and pristine benthic habitat I saw. This year, we are conducting an acoustic and video survey of the entire Oregon coast. It is a rockfish survey stretching from the Columbia River to the California border. When complete, we will have covered over 700 transects from the 80-meter depth contour to the surf line, both inside marine reserves and out. We have had to revisit some important areas due to hypoxia, so we got the opportunity to see how rockfish react to different levels of dissolved oxygen at different times of the year. What have we seen? Are they working? Again, I'm no scientist, but I am pleased to report what we have seen within the established marine reserves in Oregon waters are good numbers of a variety of fish species with good numbers of juveniles as well as some impressive sized mature adults. It would be hard to argue that marine reserves are not working. More on the drone video featured at the beginning of my comments. This video was taken by Matt Brandt as he caught us making acoustic transects on the Pacific Surveyor within the boundaries of the Cascade Head Marine Reserve. When I saw this video, I was first struck by the beauty of the ocean and the sunset that evening. It is also a very good surface visual representation of a marine reserve from the surf line to the west, in this case to about 2 miles offshore. The 3 large marine reserves in Oregon each extend to about 3 miles offshore. I was also struck at how lonely our research vessel looked out there. 10 years ago, you might have seen a few commercial crabbers or salmon trawlers in that expanse. You might have seen a few small sport boats targeting rockfish or lingcod. You might have seen a few charter boats from Depot Bay. No more. No more fishing, no more catching. No anchoring for the night or to wait out a storm. No bottom contact of any kind unless it's an emergency. What about the fishermen and other ocean users that used to frequent these areas that have been set aside? They have moved out and gone somewhere else. Some are retired, some are dead. All are aging. As time goes on and the generations change, there will soon be no one fishing who has ever fished these reserved zones. Having never been in these areas, they will have no reason to nostalgically reminisce as I have done with you today. Today, I believe the establishment of Oregon's marine reserves was a necessary step in a forward-thinking world. It did not come without pain for those who traditionally used these areas. The current threat to fishermen is not a few square miles of good reefs being off limits. I'm sure they're benefiting from the edge effect, that movement from rebounded fish populations moving out of the protected zones. They do have tails after all. No, I believe the threat comes from climate change and changing ocean chemistry. Among other effects, increasing levels of ocean acidification have the potential to hinder the production and growth of very valuable seafood resources by not allowing the megalopes or small creatures to properly build their shells, notably Dungeness crab and pink shrimp. These are 2 of the fisheries that are very valuable to the state of Oregon and the economic health of our coastal communities. I'm especially worried that vulnerable populations of ocean creatures may have bad luck with the timing of extreme levels of ocean acidification or hypoxia. Even worse, imagine multiple stressors when 2 or more levels of ocean chemistry imbalance conspire to occur at the same time. I heard this phrase, to live on the land, you have to understand the ocean. I believe this is true. We must continue to study and understand the changing ocean, for what's out there is coming here. We need to continue to be vigilant and support research and monitoring and act like our lives depend on it, because they do. Thank you for listening.
All right. Well, that was super powerful, and I want to thank again Pazar for recording that, and also to the folks at the Oregon Department of Fish and Wildlife who helped out with some of the video content for that. We're going to bring back all of our presenters that are available: Mark, Julie, and Scott, and we will up some questions. So again, keep your questions coming in the comments section, and we'll try and pull these up. And I will just start by kicking off a question here for you, Mark. This one comes from Hans, and he says, the Hsiao Bank, Stonewall, and Perpetua Marine Area has a long history of data collected for research and management. What can we do to help NOAA dedicate this area as a marine reserve under the 30/30 initiative? And I guess anybody could take that one if they wanted to.
Thanks so much for asking that question, Hans. It's good to hear from you. It's been a long time. So those 3 rocky banks, large rocky banks off the coast of Oregon, are important habitats from the standpoint of being very productive fisheries. And it makes sense that some fraction of that fishery area be used for population replenishment, like I was talking about during my talk. This 30 by 30 that Hans was referring to is a call by marine scientists and conservations around the world to protect 30% of the ocean by the year 2030. The idea being that with that much protection, there will be sufficient spillover of fish and sufficient larval seeding by these protected areas to enhance the ecological resilience of the rest of the ocean as all these big changes are coming. Okay, to get to the meat of the question, how do people move forward with that? I think probably right now with the present administration, a movement toward a national marine sanctuary process would be a very good idea for the Oregon coast. Oregon is one of the few coastal states that doesn't have a national marine sanctuary. So a portion of these banks would be valuable. I was lucky to do deep sea submersible dives on these banks back in the 90s, and they are truly remarkable places. Thank you.
All right, next question. Let's see here. This one's for Julie. Has your restoration work been coordinated at all with the restoration efforts supported by the Pacific Marine and Estuarine Fish Habitat Partnership?
Yeah, so not PMEP directly. We mainly relied on community partners like the South Slough National Estuarine Research Reserve and the Wetlands Conservancy to identify restoration projects. And we also use the Oregon Watershed Restoration Tool, which is freely available to the public and really interesting, and just identifies all of the restoration projects that have occurred throughout Oregon. Yeah, and I also wanted to mention that all of our work initially was mainly focused on salt marsh habitats. So yeah, I think there's probably more like eelgrass restoration work happening through PMAP and stuff. But yeah, I think in the future, and especially what we realize is that folks are really thinking about things on a larger scale. So for the next iteration, we're hoping to include more estuarine habitat types. Thanks for the question.
Yeah, all right. So next question here. This one is for Scott. And we see that the estuaries are becoming, increasingly more important for rockfish and other species, as you mentioned in your talk. What role, or how is that changing potentially your role in being involved with Oregon's estuary management plans or other fishery managers potentially?
Right. Yeah, I'm not directly involved in estuary management exactly, but certainly within the context of Oregon's nearshore strategy and the development of that plan and the indicator species, I've been able to play a role. And our work has also been passed along, as Julie was just talking about, this organization PMAP, to them as part of their estuary mapping plans. And it is in fact redirecting some of our considerations of how do we understand seagrass restoration efforts that, for example, South Slough and other organizations are working on, and how are we seeing changes in the fish communities as those restorations work, as another way to document that effectiveness. So it's redirecting some of the questions that we're asking in support of those conservation and restoration efforts.
Great. All right. So next question, it's a pretty smart question from Alex here. This is for you, Mark, I believe, but Scott, you may be able to field this as well. Since finding parent offspring parents is hard, what about sibling analysis? Are those analyses a good indicator of connectivity? How informative is sibling analysis compared to parent offspring?
I'll let you take that one first, Mark.
Okay. I'll take a shot at that. Sibling analysis will tell you whether or not larvae that were spawned at the same time and place actually stay together during larval dispersal and then end up at the same location. They don't tell you where they came from. So it's an important question, and it's certainly relevant to understanding larval dispersal, but doesn't really get to the connectivity issue. There's also a second part to that question about whether or not analyzing adults only would be useful. And the answer would be yes, except that there's always many fewer adults than there are juveniles. So if you want to get a big sample size, you want to sample a lot of the juveniles because you'll know if you can find their parents, you have a greater probability. Thanks.
Yeah, and I just add a little bit to that. Of course, because rockfish live so long, it's possible to have parents and offspring as adults in the same place, potentially, because just they have such long lifespans. And as far as the sibling and moving together, I don't know if it's been done on sort of our more traditional marine species like rockfish and things like that, but there's evidence to show that juvenile salmon, as they move down through the estuaries and into the ocean, and they come back a year or two later, are coming back effectively with the same individuals that they went to sea with. So definitely in that case, sibling analysis shows a lot of togetherness, if you will.
Julie, want to take a stab at that one? Or are you gonna leave that one for Scott and Mark?
Not my
I've
area.
got a question for you. I've got a question for you, Julie. This is actually mine as a recreational user. I'm interested if recreational groups or users have been involved in any of your focus groups, and whether or not they have. Do you see a role for recreational users in wetland restoration and conservation?
Yeah, certainly. We had several recreational users of restoration. We had so many different kinds of people. We had teachers, folks who just like to go out kayaking in the estuaries. Yeah. So yes, definitely we had recreational users. They can also play a huge role in restoration projects because of how important community support is to get projects off the ground. Their advocacy can really make or break a project. Also, lots of restoration projects rely on volunteers to monitor these projects over time, which is so necessary to make sure we're keeping track of how effective they are and understanding how we can improve efforts in the future. So yes, recreational end users have a huge role in restoration.
Okay, here's one that I think anybody could take this one. This comes from the infamous Mr. Engelmeyer out there. His question is, can or maybe should Oregon's marine reserves and protected areas include estuaries?
I would certainly suggest that if we're interested in the connectivity of these systems, and that they necessarily need to be included in the current network of reserves that we have, or current set of reserves that we have, but that estuaries should be definitely given special consideration for protection of those essential natural habitats if we care about fish.
Yeah, I totally agree, Scott. They're super important habitats, as Mark mentioned early in his keynote, and really highlighted everything is connected. And especially if we're thinking, long term about large scale restoration, incorporating those into protected areas would be really beneficial.
Thumbs up. All right. Okay, well, we're gonna move into the tough questions now. Then let's see. We talked a lot about connectivity tonight. Connectivity between the estuaries, connectivity between protected areas, adults and parents. And I think there's this sort of $1 million question out there about Oregon's marine reserves, and are they close enough for good connectivity for most fish species? We heard from you, Mark, that we need more marine reserves. So how many more do you think we need in Oregon, or can you even say that in the absence of maybe certain goals?
Yeah, the thing about the thing about marine reserves is, you need to have your goals laid out very clearly before you start counting things and measuring things and things of that sort. That's a social decision. But what I can say is that to get the benefits that are accrued within marine reserves, especially the augmentation of fisheries from reserves, they have to be a fairly substantial size. I've always looked at Oregon's marine reserves as sort of a test case. to show people that they work here in Oregon just like they work every place else in the world, which has always been found. So I can't say how many more or how large they should be. I can just refer back to that um, general target that's been established by a variety of people, about 30% of the habitat by 2030. Um, that's an arbitrary number to some extent, but it gives an indication of the the real size that's going to need to be protected to protect the rest of the areas that are open to fishing and other activities.
If I could add to that, Charlie, and as you know, I could have given a different talk tonight, specifically talking about oceanographic dispersal out of marine reserves, because as far as I know, we're the only ones that have ever done those drifter tracks that Mark showed out of an actual marine reserve on the Oregon coast. And we did that out of Port Orford, and we let drifters go during the spawning season for rockfish. And we would let them go, and a day later they would run aground 10 miles away. And we'd go pick it up, we'd haul it back, we'd let it go again, and 2 days later it would run aground 12 miles away. We'd go back and we'd pick it up and we'd let it go again. And we did this time and time again. And the furthest it ever went was like 26 miles, if you want to talk about how far we might expect ocean currents to disperse something, until we finally let it go in May, after all of the upwelling had started, and it took off and was on its way to Hawaii. But generally speaking, they do not, at least out of Port Orford, there was a lot of what we call local retention. And so if the goal is to have a connected network of reserves, they would have to be a lot closer together, and there would have to be more of them than there are now.
And one part of that whole issue is the fact that larvae do behave. The larger the larvae get as they grow, they turn into little fish. And what they're able to do at a fairly young age is select the depth at which they're living, which makes larval dispersal much more complex. And we're also learning that larger larvae have amazing senses with the ability to sort of smell the habitat to which they're heading or hear waves breaking. And so they have much more control than we've often given them credit for. The drifters and the virtual larvae and models are very useful for sort of defining sort of a domain over which dispersals are likely to happen. But these little animals are really remarkable at doing what they do.
Yeah, absolutely, Mark. And in some ways, we're probably with the drifters defining what the maximum dispersal space might be. And in reality, it might be less than that.
I guess I don't have much to add, having I don't think have worked as long in these areas as Scott and Mark have. But just a follow-up question. Mark mentioned some of the barriers to creating more of these reserves, in especially the process, the long and arduous and thoughtful process that goes into establishing them. Are there any other barriers that you both see in terms of public support or anything else to establishing more reserve areas?
I think Mark touched on them, and Al did as well in his talk. And even since the marine reserve process, the ocean has become a lot more complex, not from a climate change perspective, but from a use perspective. Now that NOAA is pushing aquaculture opportunity areas, and they're setting up test areas for wind energy and wave energy and so on and so forth. And our ocean is, from a management perspective, kind of being carved up, if you will, into different use areas in ways that it didn't used to be. And so to continue to also carve out space for reserves becomes, I think, socially a lot more challenging. I think just it makes it more difficult, and ultimately it comes down to people priorities, which I guess my science can't answer.
Yeah,
So we're
I
almost.
agree.
Oh, go ahead,
sorry.
Mark.
I was just going to
Finish
say, I
up.
agree with what Scott's saying. You know, it's ocean zoning is definitely coming. The ocean is being carved up. So certainly some of those pie pieces have to be areas that we're setting aside for posterity setting aside for social and cultural heritage reasons and for fisheries replenishment.
Well, we're just about out of time here. I could ask a lot more questions. I want to note and recognize that there's a lot of questions about 30 by 30. And I think we've kind of danced around some things that have to do with Oregon and that initiative. And ultimately, I think Oregon needs to settle on some goals for that. I will highlight, though, that this audience is looking to the science to community to help define those goals for Oregon and what they might look like. And certainly there's a lot of social considerations and hurdles there. But judging from the comments and questions that we had coming in, it seems there's an appetite for learning more from scientists like yourself about the connectivity and our system of current protected areas in Oregon as it relates to some of those federal initiatives. I want to thank all of our presenters, Julie for pinch hitting like last minute and dealing with obstacles with her connection, Mark for tuning back in and giving Oregon a chance again, even though you're over there in the warmer state of Hawaii. Back to your roots with Cape Perpetual. We really appreciate having you here. Scott, it's always fantastic to have you and to see you in a more academic setting rather than down on the fishing dock. Of course, I enjoy seeing you down there as well. So I want to thank everybody. I want to thank our audience for participating. I want to thank all of our organizers. I especially want to thank Tara for all her amazing work within the collaborative and bringing together all our partners, Paul Engelmeyer, Audubon Society, Joe Liebezeit, and my coworker, Bree Goodwin, who all were instrumental in sort of pulling this off. So thank you all. Thank you all for being here. We're going to wrap this up, and we have a video that we're going to show. We'll adjourn just with this short video highlighting one of the most magical places where the land meets the sea, the historic 804 Trail here in Yachats. So thank you, everybody, for being here, and we hope you have a wonderful weekend.
It has this unspeakable beauty that is so therapeutic for anyone who walks this environment. It's a point of renewal.
The 804 is a historic listed trail, and it's been used for thousands of years, and then even more recently with non-Indian population.
I like to use all of my five senses as we walk along the trail. Take a deep breath, smell the ocean air, listen to the waves.
So we're here at Smelt Sands Beach, and Smelt Sands Beach is just north of Yachats on the Central Oregon coast. So the role that surf smelt play in the marine ecosystem is that they are a trophic link. Well, this sand is just the right sort of gravel size for the eggs. And what's really neat about the eggs for surf smelt is that when they come in to spawn, there's usually like 2 or 3 males with every female. And as soon as the eggs are fertilized, they actually form outside coating that turns inside out. And then it anchors it into the sand. And so they stay in the sand and they develop, and then they hatch out. What they do is they eat the things that are really small, like copepods and isopods and larvae of even small fish. And they transfer all that energy into larger predators. They're a really important link in that food chain.
And the 804 Trail is a great place to see the marbled murrelet, which is a species that's listed as threatened. It's a seabird that has a split habitat strategy, which means that it occurs in 2 major ecosystems during the breeding season. It forages in the ocean and it flies inland to nest in the forest. This is essentially the largest reserve for the murrelet along the west coast. And that is why it's important to have this land and sea protected.
My favorite part of the trail changes with the days, the months, and the seasons. I'll see the old beaver dam, or the Oregon iris pops up in the spring. Or I go down to see the shell midden and see what our ancient ancestors created. As we walk along the trail, you can see how the trees are shaped facing towards the southeast. The wind brings the salt to the windward side of the branches, and the salt kills that part of the branch, but the other side of the tree thrives just great. So that's why they're shaped from the northwest winds during the summertime, bringing the salt on and depositing it on the branches. It's a great oceanfront trail that many people, many different modalities can access.
This whole trail system had been used for many thousands of years by indigenous people as a road, as a location for shellfish hunting and fishing. This was the impetus for greater connectivity.
The history of the 804 Trail goes pretty deep. We're right at the junction here between open sandy beach habitat and rocky shore. In the Alsea people's origin myth, our ancestral peoples of the Confederated Tribes of Siletz, Alsea and Yaquina, Yahik people, Saisla and Lower Umpqua people, those 5 groups of people are all one people, speak the same language. And so there was a lot of visitation, trade on the beach. You know, we're still here hoping that we can regain a significant portion of our homelands and still take care of those resources as we did in the past. Those are gifts from our Creator in our traditional views.
Through negotiations and mediation, the 804 South became a hiking trail as well, and that leads to the Yachats River. And then it continues on to Yachats Ocean Road, to the Amanda Trail, and into a network of 27 miles of beautiful oceanfront and forested trails at the Cape Perpetua scenic area. This whole trail system is a part of the overall Oregon Coast Trail, running from Astoria to the California border. If you're mentally stressed or physically disabled, it's a point of renewal.
Be my friend. My face to the sea and my back's to the land.
We also have opportunities for our visitors to engage, to work on the trail, invasive species control, sea star monitoring. There's cleanup on the beach. And of course, BioBlitz Community Science Project. You just download the app, which is free, join, be an observer in the Cape Perpetua BioBlitz series. And that's a database that's housed under iNaturalist, which is an international program. Literally millions of observations by community members. All of them are just a really solid opportunity to raise awareness and appreciation.
I think it's important to remember the history of how people fought to keep the 804 Trail in place. And as recreational users, I hope people are respectful of all the things they see and find along the way. Leave it as you found it or better. We all need to take care of this beachfront that we all enjoy.
My face to the sea and my